Państwo, witam Państwa serdecznie na kolejnej sesji, tym razem sesja pierwsza, naukowa, mikrobiomy i mykobiomy w badaniach naukowych. Podczas tej sesji gościmy przedstawicielkę Komisji Europejskiej, panią Magdalenę Gajdzińską, która z ramienia Komisji Europejskiej jest osobą odpowiedzialną za badania, za zagadnienia związane generalnie z mikrobiomami. Także bardzo serdecznie witam przedstawicielkę komisji i pozostałe panie prelegentki, które będą przedstawiane już przez prowadzących sesję naukową. Także życzę Państwu owocnej tutaj sesji i oddaję głos prowadzącym. Dziękuję.
Dziękujemy. Dear ladies and gentlemen, dear colleagues, it is my pleasure to chair this session together with Professor Aleksandra Obrempalska-Stemplowska. This session will be devoted to various aspects of microbiome and mycobiome. The presentation will contain problems related to ecology and ecology of plant and soil, and I'm sure that it will be very, very interesting and very fruitful session. The first keynote speaker will be Professor Gabriela Berg, our precious guest of this conference from Technological University in Graz in Austria. Let me present a short biography of Professor Berg. She studied biology, ecology and biotechnology at the universities in Rostock and Greifswald. Obtained her PhD in 1995 in microbiology from Rostock University in Germany. In 2005, she became a full professor in environmental biotechnology at Graz University of Technology in Austria. And in 2021, an additional professorship in Potsdam, Germany. Okay, let me give some information in Polish. Proszę Państwa, ten wykład jest realizowany przy wsparciu finansowym Gminy Lublin w ramach programu Visiting Professors in Lublin. Wykład w formie streamingu będzie ogólnodostępny dla mieszkańców Lublina. I would like also to point out that we will be very grateful to all speakers of this session to keep the time. When the time will be approaching, I will stand and give the sign, just let's say 5 minutes left to finish the lecture. Professor Berg, please, the floor is yours.
Yes. So thank you very much, and it's really a pleasure to be here, and thank you very much for this invitation and organizing this very, very interesting conference. And after our two plenaries about the fungi and the plants, so we will now switch to the microbiome. And I think I brought you here a picture where you can see some microbes on the root. So they're inside of the root and outside there live more than people on Earth. And I think it's really convincing. I think I will switch to that mic. So because there is a light so much coming. So yeah. And so the microbiome is really the sum of all microorganisms in one environment. So it's also the fungi and the archaea as well. So because this is the first, and so I will introduce you into the soil microbiome and the plant microbiome, but I will also go a little bit to the human microbiome as well and bring that all together in the frame of planetary health. So, as requested. And it was nicely introduced. I really enjoyed this art presentation, And for me, it was a little bit the earth, so how it develops, the evolution, but also the Anthropocene was visible, so with that disturbance which we have. And this is exactly what we have. We live in that century of the Anthropocene. And this is characterized by already being beyond the borders of our planet. So you are all aware that we have the crisis, the biodiversity crisis, the nitrogen crisis, antibiotic crisis, climate, pollution and microplastics. And everything has a direct connection to the microbiome. And on the other hand, that means as well that also microbiome management can help to cope these challenges which we have at the moment. And this is the idea of my presentation, to show you that, how we can do that. And originally, for a long time, was working on the plant microbiome until I realized that the plant microbiome is embedded in the whole microbiome circle, and that it connects us. But look on the global food losses. So we have a lot. It's for the fruit and vegetables, it's 45%, and for the roots and the tubers as well. So many people are looking on the field. So what can we change in the field that not all the fungi eat 1/3 of the world harvest as they are doing that for centuries. And there's also this post-harvest area which is much more crucial and is widely ignored. So therefore I also included it a bit into my presentation. So, and yeah, this is the visualizing of the microbiome network. And so we really realized in the last 20 years, this is a time that microbiome research is existing. It was emerging from the field of microbial ecology, and now it's really present in all different subjects. And so we realize that it's not only the soil microbiome, it's really the soil microbiome going to the plant microbiome and then reaching also our gut microbiome as well. So, and due to the importance of these connections, or I mean to look on the whole cycle is a little bit challenging, but what we can really do is to look on different connections. So, and this is also these are the sections of my talk. First the soil plant, then a little bit on the post-harvest, only to mention that this is really crucial, and here is a lot of research, and then also to the food-gut connection. So let's start with the plant microbiome, and here I have 3 examples for you. So the first, this is the alpine environment or the native plants in natural ecosystems, and I'm studying that since my time at Rostock University to understand the plant microbiome in nature and the entire functioning. Because if you go or if you start into the field, so there it's already reduced, and for example, a lot of fungi don't exist in the agricultural ecosystem. So, and then I will move to oilseed pumpkin. This is an example from Styria, where I live now, and so this is our main crop there. We are producing very good oil from this, and is very tasty and healthy. And at least I have a worst case example for you, and this is from the RSC. So starting with the alpine flowers, these are normally very nice excursions, what you can do. And this is 1 hour away from Graz in the alpine environment. And we were interested in looking into the seed microbiome of these alpine plants. And this is a very beautiful and highly diverse meadow. And of course, we expected that these plants, here are the ones which we studied, they grow together there for centuries, and they will share a lot of the microbiome because it's the same soil. So, and when we analyzed them, we were really surprised because what we found is, first point, an incredible diversity inside of these very, very, very small seeds. So it was challenging to sample these plants. They are very small, and the seeds even smaller. So altogether we found around 12,000 different OTUs, which is a bit microbial species, but they shared only 11. So and this we have not expected, that it's really so unique, and that these seed microbiome also contribute to soil microbial diversity, because the soil in terrestrial ecosystem is really the exoskeleton and the seed treasure chest for all microbes. So, and it was similar for fungi and for archaea. And we were really surprised also when we looked on the list of the fungi, because this was like a European list of plant pathogenic fungi. So they are all associated also with the seeds of these native plants, but for sure in nature they have a completely different function. For example, degrading the seed shells. So, and it's the reduced biodiversity and all these anthropogenic impact what makes them then to the pathogens. So, and we could also visualize the microorganisms inside of these seeds. I'm always enjoying also this visualization, because it convinced sometimes much more than to see only the DNA, although it can be beautiful, as we have seen. So, and there is also a proof of principle what we have done here, because these Gentiana seeds, they were germinate under gnotobiotic conditions, and you can see that the root part is here colonized by a highly diverse microbiome. So, and this is what we learned, that there is a vertical transmission of approximately 50% of the microbiome, and then there is a horizontal acquisition from the soil microbiome. And this makes a lot of sense, transporting the beneficial microbes from one generation to the other, and on the other hand also to adapt the plants to the new environment. So it's to be honest, it's like for human as well. So and we figured out it's now for all organisms. But looking on the conclusion, I mean, if you look on our crop seeds, which are sterilized, which are produced only on specific sites in the world and then stored for a long time, they have a much reduced diversity. And this is what I would like to show you now. So the impact also of breeding and the impact of fertilization and agriculture on the seed microbiome. So, and therefore I selected these pumpkin, because I mean, normally all our crops, they are domesticated for a very long time, and it's very difficult also to find out the breeding lines all over the world. So, but here, this pumpkin, it was a mutant, and it was found 100 years ago in Styria, so the place in eastern Austria, not that far from here. So, and the farmer was very intelligent, clever farmer, and he thought maybe I can do something with that, because the pumpkin lost the wooden seed shells. So, and what he has done then is he roasted it, and then he produced the oil, and then he thought it's really good. And so we have here only a domestication and breeding period for 100 years. And we have a cooperation with these breeders, and so we have a clear breeding line, and we have all the genetic information from the plants. So, and then we studied the seed microbiome, the rhizosphere microbiome, and the soil microbiome. And what we found out is, first of all, again, here I would like to start with the microbes. This is all, these three pictures, it's inside of the seed, and you can see how the microbes are sitting and surrounding the cotyledon, and then colonizing also the seedling. And interestingly, what we found out is, as higher the diversity within the seeds, as more resistant they are. So, and this was a surprise, and it also surprised the breeders a lot. But due to the fact that it's such a small company, and they are well educated by our university, so they start really microbiome assisted breeding. So, and this is a summary. So what we found out for the breeding, because normally the breeders, they are looking for big fruits, big crops, so for high productivity, and also they look for resistance. So, and there are a lot of examples now. I mean, these are from our groups, but there are much more from other groups that, for example, if you breed for resistance, then you also pay a price of diversity. So, and there's always a correlation between the microbiome, because the breeders shaped much more the microbiome than the phenotype of the plants. So this was also a surprise. And I would like to bring your attention to the cancer. This is also a new perennial grass. So the future of agriculture, we hope that we can integrate much more perennials, because they are more sustainable. And they have now 10 breeding cycles of that cancer. And now we have seen that in parallel they reduce the diversity of the seed microbiome. So it will have the price, the larger seeds, that they are then more susceptible to pathogens. And I mean, the conclusion is, of course, that we have to consider the holobiont, not only the plant, it's both. It's a plant microbiome and the co-evolved thing. So, and my last example, this is a worst case example. This is an RLC, which disappeared in the last 30 years. And so there were a lot of restoration efforts from international NGOs, but all of them failed. But fortunately, there are some plants. One survived. But these worst case scenario or environmental disaster, is a product from intense agriculture, because the former Soviet Union decided to switch the desert into intense cotton production in Uzbekistan and in Kazakhstan, and they used the river of the Amudaya and Surdaya for intense production, and applied a lot of pesticides, so that these dried out RLC basin is really full of the residues, and the people living there, they are really sick. So, and we thought it would be interesting to look on the plant microbiome, how it looks like, because it's also a little bit a future scenario of our planet. So, and this we have done, and we used also a succession gradient of these Zoeda acuminata. And we found very, very clear results, which I have not expected. So in the early revegetation, so there were only archaea fulfilling the function of the plant microbiome. I think this was one of the first examples showing that the plants can survive by archaea, but it's much more difficult for them. And under better conditions, then they are completely replaced by the bacteria. So the ecosystem is restoring. And this study, it was also in parallel, it was our first study on the viruses. So I think they also belong to the microbiome, and they are the drivers of the microbiome. And we could clearly show that there are very, very clear pattern of these viruses. So they are following, and there was a complete shift. So, and they have also shaped a lot of the plant microbiome. So I think the viruses, they are drivers of the plant microbiome, which we have really to consider. So, and this is a summary of the first, and this was also the longest part of my talk. So what we see, the signature of the plant microbiome in the Anthropocene is a shift from plant beneficial bacteria to plant pathogens. So the diversity decreased, the evenness decreased, and the specificity decreased. This is an opinion paper based on a lot of publications. It's not only ours, so there is a lot of other work. And so we have more dysbiotic microbiomes. We have a prevalence of fast-growing organisms, the so-called R strategist, and we have a lot of hypermutators, and we have increasing antimicrobial resistance. And this is similar also to the human microbiome. So I think we have really to look on the whole microbiome and see how it changed. So, and there's always a question, what is a healthy microbiome? And this is, I think, now clear that it's the functional diversity is what matters. So the functional diversity of the plant microbiome, and this is also the same for the human microbiome. So what we need to avoid all these pathogens, these multi-resistant pathogens which emerge everywhere, is microbiome management. So a very short excursion to the post-harvest microbiome, because here we selected the apple, and this was a citizen science project with kids. So, and we thought we have to convince them that the microbiome contains a lot of beneficial bacteria. So it's the shift from considering bacteria and fungi and archaea as pathogens to that the microbiome means health. So and this was what we have done with the kids, and so we were also surprised about the results because we have not expected that high diversity in the pulp. So, and then we also studied the impact of storage, and in that global agriculture, which was definitely a mistake because the apples, they are stored for a long time. It's 1 year until they go to the supermarket, sometimes 2 years. They look the same, but the microbiome is not the same anymore. And so we have even a storage-specific resistome. So, and there is increasing, there are in the clinical relevant antimicrobial resistance genes are increased. So this is the only possibility to determine the age of apples is to analyze the microbiome, because it looks completely the same. So, but this, I think, because we are eating that, is really important to consider. And in that post-harvest period, also there are much more chemicals applied than in the field. And so, but most of the people are looking or try to make this part more healthier and sustainable. So, is that really important for us? Therefore, there is a new research direction emerging, and this is the exposome research. So what the people from medicine saw that, I mean, of course, within the genome there are some marker, and there are crucial points to predict diseases, but it's much more the environment. So, and therefore they established also the cohorts. So these are children, then 1,000 babies born, and then they are looking for them during their whole life cycle. So, and there are these very big EU projects now studying these cohorts and try to figure out the impact on environmental factors on the microbiome, on human health. So, and it's our turn to be involved with the microbiome. And so the first step was that we were looking for the food microbiome, and we thought the apples were really ideal to figure that out. And there's always the argument, nah, but we cook that, and the microorganisms, they cannot survive, so there's a stomach pH of 2, but now we know it's colonized, and as endophytes they survive well. So, but they also survived the shredding, the boiling, and the preserving, even the drying, although we see a shift. So, but in the sterilized food, and this is for example, now the parents giving to their kids these squetchies, or don't know how it's named in Poland. So, and think that they are doing something good. But the kids, they really need also the microbiome, because in the first 3 years they assemble the microbiome, and this is really crucial that they get enough diversity from the environment. So, and it is very challenging to show that really these plant-associated microbes establish in the gut, and they can survive. This is really challenging to do that, because you have to convince then the people to eat for a certain period of time only this, but they are also different, because one is doing exercise and the other is doing something else. So, and therefore we have done here, we analyzed all the metagenomes which are available in the public data set and ours as well. So, and the first point is there is a significant overlap of the human gut microbiome and the food and vegetables. This is the inner circle. So in pinkish, this is the humans, and the food and vegetables is in blue. So, and we could show by this analysis that it's 2.2 on average of the proportion which is originating from foods and vegetables. But it's even more convincing to analyze the data from the American Gut Project. So this is a very, very large data set. It's also, I think it's the biggest citizen science project which we have, and the people, they give everything, also how much veggies they are consuming. And so the vegetarian and the vegan people, they have significantly higher plant-associated microbes in their gut, up to 30%. So they are really, really crucial. So, and this is then the last summary, is that there is that connection. I hope that I convinced you, from soil to the plant, and then during the post-harvest period we have the change, and then also to the human. And this we have to consider also if we are doing something for agriculture, that we are eating that. And so it's not only the big pumpkin, so it's also the adipositas, which is resulting from that. And there is a lot of evidence that there is exactly this link. So, and what we have to do is biocontrol, of course, but also microbiome management. There are very good examples for that. Long-term examples like fecal transplant from the Chinese medicine since 3,000 years. It's until now very successful, success rate of 96%. And the interesting thing is that also here it is completely in parallel. So if you want to control the health of a plant, of an animal, or of humans, so you have to change the abiotic conditions, or you can add the microbiome, single strains, or whatever. So, and as one example, so we have also a long-term collaboration with a sugar beet seed company. And so what we have done for them is to develop a portfolio for healthy sugar beets. And it started from protection towards abiotic stress, biotic stress, until post-harvest. But it's even much more what we have to do. So last year, we, together with 40 other co-authors, we published that article. And these are ecologists working with wildlife and corals and natural ecosystems. And before of that, I was not aware how sick our ecosystems are, but we have seen it also with the COVID, with the bats. So if the pressure is the abiotic pressure is too high, and the biodiversity, the microbial diversity is going down, so then we will have a lot of outbreaks in the future. And it's not only humans, it's not only our crops, it's also our ecosystems and the organisms. And therefore we have to understand this. So, and I think there are a lot of teachers here, and what we have done is a MOOC, so this is an open access online course, and it's we are using it for teaching our students to the microbiome. So there are 6 chapters, 6 lessons, but you can also do it yourself or give it to your kids. So, but I would like to advertise this a little bit. And this is my last slide. So it's, of course, it's not my work alone. So it's a whole group. And this was last month in May. So there were also some colleagues with us, and I'm perfectly in time. Thank you.
Thank you very much, Professor Berg, for a very interesting and inspiring lecture. However, I'm very sorry, but we don't have time for discussion. So I invite everybody or interested people to find the time during the break. Thank you very much.